Welcome, medical students, to this Masterclass Explainer. We're diving straight into Henry Windle's 2025 to 2026 SF Medicine MDU 2207 course notes. Our goal today is absolutely clear: strict, deep, exam-level retention of molecular mechanisms. We're going to go slow and deliberate. We'll break down the cause, the mechanism, and the consequence of every single process so you walk away with a true systems-level understanding of panomics and precision medicine. Let's get right to it. Okay, so here's our roadmap for mapping the central dogma. We'll start with an introduction to personalized medicine, move through genomics and epigenomics, then hit transcriptomics and spatial context. After that, we'll cover proteomics and phosphoproteomics, zoom out to metabolomics and microbiomics, and wrap it all up with multiomics and the clinical future. All right, Section 1, Introduction to Personalized Medicine. I really want you to anchor your understanding on this exact definition from the National Cancer Institute. The main goal of personalized medicine isn't just about treatment. It is to prevent, to diagnose, and treat disease by using highly specific information about each patient's genes, proteins, and environment. So why do we need all these omics technologies to do this? Well, it's because comprehensive molecular profiling of genomes and proteomes gives us a much deeper understanding of the underlying disease mechanisms. And that depth of understanding is literally what underpins this entire shift toward personalized medicine. Moving right along to Section 2, Genomics and Epigenomics. Starting with the static code, genomics is the fundamental layer here. It maps a patient's static biological blueprint, and we pull this off primarily through two methods: whole genome sequencing, or WGS, and whole exome sequencing, known as WES. Now, looking at the clinical applications, genomics is absolutely critical for identifying monogenic disease variants, single nucleotide polymorphisms, those are your SNPs, and general genotypes. It's also the foundation for pharmacogenomics. A really vital example for your exams is identifying CYP450 variants, which dictate whether a patient is going to have a slow or rapid metabolism in response to certain drugs. It also drives cancer genomics and targeted therapies, specifically using markers like EGFR and BRCA1 and 2. But we have to talk about the limitations. There are two primary hurdles you absolutely must memorize. First, the frequent discovery of variants of unknown significance, and second, the massive interpretation challenges that pop up when you're analyzing these gigantic genomic datasets. So if genomics is the static code, think of epigenomics as the system of modifiers. Let's define it clearly. Epigenomics is the study of heritable changes in gene expression that happen without altering the underlying DNA sequence itself. The main mechanisms you need to know here are DNA methylation and histone modification. Clinically, profiling these cancer-specific epigenetic patterns is a total game changer. It lets us classify tumors, spot distinct cancer subtypes, and establish specific biomarkers for early disease detection. Plus, in developmental biology and aging, epigenomics is used to map how cells differentiate during development and to actually track cellular aging through what we call epigenetic clocks. This perfectly illustrates the cause, mechanism, and consequence of epigenomics in disease. Step 1 is the cause. Environmental factors, like smoking or diet, trigger changes. Step 2 is the mechanism. These factors shift reversible gene expression patterns. And step 3 is the consequence, which is a clinical goldmine. Because these epigenetic changes don't alter the actual DNA sequence, they are reversible. And that reversibility means they are prime targets for new therapies. A perfect example of this mechanism in action is using inhibitors that target DNA methyltransferases. Next up, Section 3, Transcriptomics and Spatial Context. Transcriptomics is the study of mRNA expression using RNA sequencing. We use next-generation sequencing, NGS, to profile the entire transcriptome. And remember, this includes both coding and non-coding RNAs. The absolutely crucial advantage to lock in for your exam is that transcriptomes captures dynamic changes. Unlike the static genome, it tells us exactly which genes are actively switched on or off at a given moment. In the clinic, this dynamic signature is used in tumor and disease classification, like figuring out if a tumor is responsive or resistant, and for understanding host immune responses in infectious diseases. Now, we have to carefully contrast how we actually gather this data. Let's compare bulk RNA sequencing with single-cell RNA-seq, or scRNA-seq. Bulk RNA sequencing averages gene expression across all the cells in a tissue sample. But because it averages everything, it totally obscures cellular heterogeneity and lacks any spatial context. It's like blending a fruit salad and trying to taste the individual grapes.
To fix that averaging problem, we use scRNA-seq, which involves dissociating individual cells from the tissue. This gives us high-resolution analysis of individual cell types. But that dissociation comes at a severe cost. You completely lose positional information, cell-to-cell interactions, and the overall tissue landscape organization.
That is exactly where spatial transcriptomics comes in, to bridge the gap. It integrates molecular profiling right with the spatial tissue context. It analyzes gene expression while maintaining tissue organization within its native environment. This actually lets us visualize which RNA is being produced by different cells in a specific tissue section. And by doing that, it eliminates vital molecular interactions between tissue components, revealing things like how a tumor interacts with its surrounding microenvironment and mapping the exact locations of immune cell infiltration within those tissues.
To really nail down a precise clinical correlation, let's look at hepatocellular carcinoma, or HCC, to understand intrasample heterogeneity. In an HCC analysis using spatial transcriptomics, we can see step-by-step how different regions behave. We find that the immune-rich tumor region highly expresses immune-related pathways, specifically pathways C and D. On the flip side, we can see that proliferation and metabolic pathways are enriched across the tumors of non-responders. This perfectly proves how spatial context predicts therapeutic response based entirely on the microenvironment.
All right, Section 4, Proteomics and Phosphoproteomics.
Next, we dive into the actual functional machines of the cell and their molecular switches. Proteomics is the analysis of the full protein complement of a cell or tissue, heavily relying on mass spectrometry, or MS. Its main applications are in biomarker discovery, identifying drug targets, and studying post-translational modifications, or PTMs. But you really need to be aware of the distinct clinical challenges here, namely the immense complexity of proteins and really high sample variability. Let's narrow in on a highly specific and critical subset, phosphoproteomics. This is a massive pillar in 2025 precision medicine. It investigates PTMs, specifically phosphorylation, to find active signaling pathways and drug targets. It drives targeted cancer therapy by pinpointing actionable kinases that drive tumor progression, which helps us select the appropriate inhibitors. Crucially, it also helps overcome resistance because phosphoproteomic profiles can reveal the alternative pathways that get activated when tumors develop resistance to initial treatments. Furthermore, it enables patient stratification. It helps classify diseases into subtypes based on active signaling networks, rather than just static genetic mutations, which dramatically improves prognosis. A vital example to memorize here is EGFR. The rapid assembly of multi-protein complexes around EGFR provides context-dependent branching points in the network. And remember, kinase specificity within these networks is primarily structurally determined.
Moving out now, Section 5, Metabolomics and Microbiomics. Now we zoom out from the proteins to the metabolic outputs and the external inhabitants that influence our biology. Let's systematically grid out these next two layers. First, metabolomics. This is the study of metabolic products in cells and tissues. It profiles small molecules to understand metabolic shifts, giving us direct insights into disease status. Its absolute supreme strength is that it reflects the real-time physiological state. Key applications to remember are early disease detection, monitoring treatment response, identifying cancer biomarkers, and, crucially, neonatal screening for genetic disorders. Right alongside this is microbiomics, the study of microbial communities in the body. Its clinical importance literally cannot be overstated. The gut microbiome directly influences inflammatory, metabolic, and specifically, psychiatric diseases. Plus, the microbiome heavily influences drug metabolism. Because of this, microbiome-based diagnostics and therapeutics are a massive growth area right now.
Finally, Section 6, Multi-omics and Clinical Future. Now we synthesize all of these individual layers into one whole, systems-level understanding. Just to grasp the sheer scale of multi-omics integration, take a look at the Human Cell Atlas project. The goal here is to map all of the thousands of distinct cell types in the human body. This emerging atlas is completely transforming how we understand disease and immunity. To date, this incredible project has mapped over 100 million cells from 10,000 people. It's truly mind-blowing.
Observe how this builds incredible statistical and predictive power in the clinic. By measuring as few as 5 to 20 proteins in blood, we can now predict the risk of developing a diverse range of 52 different diseases.
These proteomic signatures perform demonstrably better than the standard tests used in clinical prediction today. They beat clinical risk factors by significantly improving likelihood ratios, or LRs. And remember this definition for your exam. LRs represent the likelihood that a high-risk test result would be expected in an individual who will go on to develop the disease in question within 10 years of follow-up. So the really crucial takeaway from this masterclass explainer is that while challenges certainly remain, the integration of these unique and complementary omics layers is rapidly accelerating clinical translation. As cited perfectly in this 2026 reference from Godardi in the European Journal of Internal Medicine, multi-omics creates a true systems-level understanding, and these omics technologies already completely underpin the shift toward personalized medicine.
I'm going to leave you with this final thought to consider. If multi-omics profiling maps out our exact probability of future disease with molecular certainty, literally down to the cell, the protein, and the metabolite, how will this unprecedented foresight change the way we live today? Mastering these omics mechanisms gives us the ultimate blueprint. Study these pathways, memorize the mechanisms, and you will understand the future of medicine. Good luck with your exams.